Hi, welcome to Perspective. I will present you a theoretical background of COVID vaccine the concept developed by Moderna. Or they fixed this mRNA inside that cargo, and this mRNA codes for the spike protein. And as you know, this spike protein is the outermost envelope of the virus. And this vaccine that they developed has passed through the clinical trial phase 1, where 8 of the people developed neutralizing antibodies against the virus. This clinical trial phase 1 is designed to learn about the safety, efficacy, and doses of the given formulation or the vaccine. Out of 8 people, 3 patients who were given 250 microgram of the injection, they developed fever and flu-like symptoms. The reason for that might be different, but the trial was itself very much successful, in producing neutralizing antibodies. Certain things they modified to develop the vaccine. Suppose they modified the bases uridine. They did it because to train the body, the immune cells cannot attack the mRNA itself. Otherwise, the mRNA can get destroyed. That's why they had to modify the bases. And some other bases are still modified, so that the unfolding of the mRNA inside the cells, immune cells, becomes much more easier. And they wrapped it in the cargo of lipid. They use the lipid because inside the cell, the lipid soluble particles can go inside the cell, and water cannot go inside the cell. So this is a major advantage to use a lipid nanoparticle. But the thing is, the lipid nanoparticles should be biodegradable as well. And the scientists are thinking about it lately, and also they have come up with a new theory, they can produce some sheets where they can weave the mRNA in the sheets and then shed it inside the body, so that later after the work is done, the sheets can destroy themselves automatically. So the problem of toxicity always remains in such cases. Like immune cells, they can take the mRNA itself. The lipid nanoparticles fuse with the cell, and suppose macrophages, and RNA comes out, and hopefully, or it has been shown that theoretically it is the protein is produced, and the protein should be folded in a proper manner as well for the right spike protein. And the antigens, the spike protein antigens, are presented outside the cell. And this is taken by the naive T cells. In a serial cascade manner, beta cells become active and antibodies gets produced against the spike protein. So in a sense, the hands of the virus, the spike protein, gets stuck by the antibodies, and the body system destroys the virus particles. The Moderna named this vaccine is mRNA 1273, and it has passed through second clinical trial phase as well, I suppose, and they are trying to bring this vaccine by the later of this year. Several of the companies and university itself, for example Oxford, Sinovac, are also in the race. But the basic concept of today's episode is to go into the theoretical aspect of vaccine development. I think I gave certain ideas to the viewers of this channel about the ideas of vaccine development against the COVID. I hope to bring some new concept in the future days.